All right, welcome back to The Explainer, everyone. Today, we are taking a seriously deep clinical dive into something absolutely fascinating and honestly incredibly complex. We're going to be looking at hereditary autoinflammatory diseases. Essentially, we're exploring exactly what happens when the innate immune system, our body's absolute first line of defense, completely misfires. So if you've ever wondered how just the tiniest genetic tweaks can unleash these massive systemic inflammatory cascades, well, you're definitely in the right place. Let's get into it. Okay, let's look at our roadmap for today. We're going to cover 1. Immunity defined, 2. MFF, 3. Hereditary recurrent fevers, 4. Vasculopathic diseases, and finally 5. Targeted therapeutics. Starting off with Section 1, immunity defined. So first things first, we really have to draw a hard line between these two distinct systems. Autoimmune diseases, think lupus or rheumatoid arthritis, are driven by the adaptive immune system, right? They involve those autoreactive T and B cells, and of course, the production of autoantibodies. But autoinflammatory diseases, they are an entirely different beast. They're driven by the ancient innate immune system. Here we're talking about myeloid effector cells, so things like neutrophils and monocytes, and germline-encoded pattern recognition receptors that don't rely on autoantibodies at all. When we're dealing with autoinflammation, we're looking at a system that is literally hardwired to respond to danger. But in these patients, the alarm system is basically just stuck in the on position. And the intracellular signaling pathways we're dealing with are frankly mind-bogglingly complex. These innate immune pathways are constantly 24/7 scanning the host cell's cytoplasm for any microbial danger signals. It's this incredibly intricate web of sensors and triggers. But what happens when just one of these nodes is genetically altered? Well, the entire cascade triggers inappropriately, and that leads to massive, unprovoked systemic inflammation. Right at the very heart of this aberrant signaling is the main cellular culprit for our entire explainer today, the inflammasome. So, an inflammasome is a large cytoplasmic complex of intracellular proteins. Now, under normal physiologic conditions, its job is to link the sensing of microbial products to the proteolytic activation of inflammatory cytokines. And it does this specifically through an enzyme called caspase-1. Caspase-1 then goes ahead and activates interleukin-1 beta and interleukin-18, which triggers a robust, healthy inflammatory response to fight off a pathogen. But the real problem is that in autoinflammatory diseases, this inflammasome activates without any true infection present whatsoever. Moving on to Section 2, FMF, which is really our prototype disease here. Let's step through the exact pathophysiology of how this works. Familial Mediterranean fever, or FMF, is driven by mutations in the MEFE gene, and that gene encodes a protein called pyrin. Now, importantly, these are gain-of-function mutations. They effectively lower the threshold required to activate the pyrin inflammasome. So think of it like falling dominoes. Step 1, you have the MEFE mutation. Step 2, the pyrin inflammasome activates way too easily. Step 3, this triggers that caspase-1 enzyme we just talked about. And finally, Step 4, an unchecked massive release of IL-1 beta and IL-18. This resulting cytokine storm is exactly what causes the devastating clinical attacks we see in FMF patients. So what does this cytokine storm actually look like clinically? The features of an FMF attack are very, very distinct. The attacks typically last about 1 to 3 days, and patients suffer from extreme polyserositis, which means agonizing inflammation of the serosal linings like peritonitis in the abdomen or pleurisy in the lungs. You'll also frequently see acute monoarthritis, often hitting the knee, ankle, or hip. Now, a highly unique identifier you absolutely need to memorize is the erysipelas-like erythema. It's this raised red rash that usually pops up on the lower leg or the foot. But listen, the most severe, life-threatening risk of FMF, without a doubt, is the development of systemic AA amyloidosis. If left untreated, that can lead to catastrophic kidney damage. Now, you might be wondering, why on earth would a gene mutation that causes such horrific inflammation be so prevalent, especially in Mediterranean and Middle Eastern populations? Well, let's take a quick, fascinating evolutionary breather here. Research actually suggests that these specific FMF founder mutations provided a massive ancient survival advantage. The hyperreactive pyrin inflammasome likely made these individuals highly resistant to certain pathogens, specifically the bacteria responsible for the bubonic plague. So kind of wild, right? A genetic trait that literally saved populations from the Black Death centuries ago is now misfiring as a chronic autoinflammatory disease today. Okay, Section 3, Hereditary Recurrent Fevers. Now, what's really interesting here is how we differentiate these rare fevers based on their genetic driver and their attack length. In TRAPS, which is caused by mutations in the TNF-SRFA gene, the attacks are brutal and often last longer than 7 days. Then you've got HIDS, or mevalonate kinase deficiency, caused by the MVK gene. Those attacks usually last 3 to 7 days. And finally, CAPS, caused by NLRP3 mutations, where the fever and inflammation can last anywhere from just a few minutes to being entirely continuous. So the actual timing of the fever, that is a massive clinical clue. Let's zoom in on CAPS for a second, the cryopyrin-associated periodic syndromes. This is such a brilliant illustration of how different mutations in the exact same gene, the NLRP3 gene, can lead to vastly different severities of disease due to that constitutive IL-1 beta release. At the milder end of the spectrum, we have FCAS, where patients develop an urticaria-like rash and fever triggered by something as simple as being exposed to the cold. In the middle, there's Muckle-Wells syndrome, or MWS, which adds progressive sensory neural hearing loss into the mix. And then, at the absolute most severe end is NOMID, characterized by chronic aseptic meningitis and severe joint overgrowth, or arthropathy. It is a stunning example of how just a single inflammasome gene can dictate such a wide clinical spectrum. Section 4, Vasculopathic Diseases. This group includes conditions like the recently discovered adult-onset VEXAS syndrome, which is notable for vacuoles in hematopoietic precursors and its overlap with relapsing polychondritis, and also our next focus, DATA2. A really prime example here is deficiency of adenosine deaminase 2, or DATA2. This one is caused by a recessive founder mutation that's particularly common in the Middle East. It classically presents with early-onset lacunar strokes, livedoid rashes, and a systemic vasculitis that looks incredibly similar to polyarteritis nodosa. It just perfectly demonstrates how innate immune dysregulation can actually target the vascular endothelium specifically. And finally, Section 5, Targeted Therapeutics. Let's talk treatments. The real beauty of understanding these exact molecular pathways is that we can now deploy highly specific, targeted therapies to shut them down. For FMF, daily oral colchicine is the absolute gold standard. And not only does it prevent the acute attacks, but it is literally life-saving because it prevents the development of that fatal AA amyloidosis we talked about. For diseases driven directly by IL-1 beta, like the CAP spectrum or refractory FMF, we use IL-1 antagonists, so drugs like anakinra, canakinumab, or rilonacept. They effectively shut off the inflammasome's end product. And for TRAPS, which involves the TNF receptor misfolding, we use TNF inhibitors like etanercept. But, and this is absolutely crucial, there is a high-yield clinical pearl regarding TRAPS that you have to know. While etanercept, which is a TNF receptor fusion protein, is very helpful, you must never, ever use monoclonal anti-TNF antibodies like infliximab or adalimumab for TRAPS patients. Counterintuitively, these monoclonal antibodies can paradoxically trigger and exacerbate the inflammatory attacks. It's a vital, highly specific pharmacological distinction, and it stems directly from understanding the exact misfolding mechanics of that mutant TNF receptor. We've covered a tremendous amount of incredibly complex immunology today, but I want to leave you all with this final thought. Could understanding these super rare genetic inflammasome diseases actually unlock treatments for everyday conditions? Well, as it turns out, the exact same innate immune pathways, specifically the NLRP3 inflammasome, are now known to be the primary drivers of inflammation in exceedingly common diseases like gout and even the endothelial inflammation we see in atherosclerosis. By studying these rare genetic misfires, we are literally unlocking the secrets of the innate immune system that apply to every single one of us. Thank you so much for joining me on this explainer, and keep questioning how the microscopic mechanisms drive macroscopic disease.